Okay, so I have to thank Nacho for basically introducing also my project. I would like to thank you guys for listening and inviting me to talk about the project that I did during my PhD in the Furlong Lab at the EMBL in Germany, in Heidelberg. Now I am at the Columbia University in the lab of Stavros Lombardis. Okay. Nacho talked a lot about variation, and we know that variation in gene expression has been studied in the last 20 years. We started with the yeast to move then to plants, mouse, flies, as you have heard, and humans. Like genome-wide association studies have been helped us in order to identify causal variants or associated variants to particular complex diseases, such as schizophrenia, just because it was named previously, but many other diseases. And now, what these GWAS studies do is to identify expression quantitative trail loci, or EQTLs, that are those SNPs that are associated with a variation in a specific phenotype. And this phenotype is the measurement of the variation in gene expression. And here is like a study done in humans where they measured gene expression through RNA sequencing of different individuals from different heritage. And what they see is that there are 3 different individuals with 3 different groups with 3 different haplotypes, and this causes a variation in gene expression. Now, this can be acting in cis or trans, as we know. Cis are those EQTLs that affect the expression of the closest gene, while trans are those EQTLs that are affecting genes that are far away. In other chromosome, in the same chromosome or in another chromosome, like easy to think about a transcriptional cascade where you have you're affecting the expression level of a transcription factor, so that all the other downstream target genes are affected as well. Now, developmental biologists believe that the development and embryogenic development, embryo development is very, very stereotyped, so that if you change level in gene expression of crucial genes, then you have a mutant phenotype. And this is like the case of some example, in particular, like here, twist or snails, that are genes that are involved in the Drosophila development. And if you overexpress twist all over the embryo, you have the formation of 2 mesoderm instead of 1. So what it is believed that development is stereotypic and variation causes mutagenesis, cause mutants phenotypes. But we were not convinced about it, so we started this project trying to answer a couple of questions, or to evaluate the extent and impact of genetic variation during during developmental stages. And we wanted also to assess the effect of this variation on transcriptional and post-transcriptional regulation. So what we did, and this Nacho mentioned before, that was extremely painful for us, is to collect embryos at 3 different Drosophila embryos at 3 different time points. And we chose to use this genome panel that Trudy McKay made available for the entire community. She sequenced nowadays it's more than 200 isogenic lines, meaning that she embred them for 20 generations in order to obtain the highest possible homozygosity for every SNP in the genome, and to decrease the block or linkage disequilibrium, as Nacho explained before. And this is extremely helpful for us, as you will see later on in the talk. So we chose 3 different time points, when cells are multipotent, when cells are specifying, and when cells are differentiating. And we are using the Drosophila organism because you can think that we cannot definitely do in humans or doing in other more complex organism like mouse. It's going to be a little bit more complicated than it is already it is painful for Drosophila for 80 lines. I cannot imagine for mice. So in order to do this, so we extract RNA from these lines, and we did, instead of doing cage as Nacho did, we did 3' Tag-Seq, meaning that we sequence only the most 3' region of the mRNA. And here is shown a representation of our data. So in the Y for each stage, you have all the 80 lines. And in blue is represented the coverage of each alternative polydenylation site. And you can see like one gene that is very known, ELAV, that has multiple 3' UTR ends. You can see that there are multiple here, and you can see an elongation during developmental stages as shown before in previous data of the literature. Another thing is that we see also some other genes that do not change at all. This protocol is extremely quantitative and wanted to be extremely quantitative, and it's giving us a single base pair and the resolution of the transcription end site. So it was pretty useful for us to call EQTLs or QTLs in general. So in order to confirm that we collected the right RNA at the right stage or for the right embryos, you can see in the paper there are many. We did multiple level of controls, but one of these, just to exemplify, is to compare the expression, the genome, like the expression profile of each one of these lines with some data previously published by the ModEncode project, that is a RNA profiling of stage collected embryos. And we can see that all our embryos are located in a specific time window that corresponds with the stage that we wanted to. So we use this, and I don't have, for timing reasons, I won't talk about the cis and trans detection that we see, the variance decomposition that we did, but we saw major events happening through trans EQTLs and the effect of maternal gene, and you can read this in the paper. But we use this data from these 80 lines in order to identify the transcriptional and post-transcriptional effect of this structural variation and single nucleotide polymorphism on a 3 different level, or for 3 different phenotypes. We wanted to see if SNPs or EQTLs that affected change in the expression level of the entire transcripts, or specific polydenylation isoform and changes in the 3' UTR. So this is a work that was in collaboration with Ewan Barney at DBI, and in particular Niels, a super talented PhD in his lab that did all the computational analysis in the paper. So using Limix, that is this algorithm developed by Paolo Casale and John Oliver, sorry, not John Oliver, Oliver Stiegel at DBI, we saw we identify a bunch of common and stage-specific QTLs and 3I QTLs, so QTLs that affect the expression level of the gene or a specific polydenylation isoform. And we were able also to identify polymorphism or QTLs that are specific to stages, to 1, 2, or 3 specific stages. So I want to talk first about the EQTLs, and I told you that we described the stage-specific QTLs, where there are EQTLs that are specific to one stage or to two stages, or in a combination of them. Now, we argued and we asked the one main question is how reproducible is or how useful is the 3' Tag-Seq in comparison to RNA-Seq? Because you can imagine, you guys know better than me, that mappability for short reads, it's a big problem for EQTL studies. So what we did is to sequence 22 of these lines at the 10 to 12 at the latest time point, and we did RNA-Seq on them, and we compare the difference in normalized mean from the level of RNA-Seq per genotype towards the effect size that we call in the 3' Tag-Seq. And what we see is that there is a good correlation, as you can see in this plot, there is a good correlation between these two data. This data set. So we validated in a certain way, and in a mappability, like after all the mappability filter and the computational controls that Niels did, that our EQTL call is solid. And here is just to show you an example. We look at the GSTD1 locus, and you can see that we tested, I forgot to mention, but we tested 100 kb, more than 100 kb region SNPs around each gene. And what we see here is a Manhattan plot where we have a single, very single specific EQTLs that is highly significant or highly associated with the variation in gene expression. And this is, as Nacho mentioned, is like a huge potential of the Drosophila system, this Drosophila system, that allow us to pinpoint in 50% of the cases in a single base pair, like a single variant that is affecting variation in gene expression. In comparison to human study, where you have large blocks of linkage disequilibrium, where hundreds of SNPs having the same p-value could be the causative SNP. So identifying the causal variation is very hard. In this case, it was pretty easy because of the short linkage disequilibrium, or the small linkage disequilibrium box. So in this panel, what you see is and in these two panels, you see the coverage per each line for the 80 lines, having the major in red, the major allele, the more frequent allele, or the minor allele, the less frequent allele. And you see that there are huge differences between these two sets. And this is confirmed also by the RNA sequencing. On top of this, we confirmed this by in situ hybridization. We took 2 lines per genotype, 2 for the major and 2 for the minor, and we saw that in the major we had expression in the MIGAT for GST D1 at stage 11 and 13, but when we look at the minor allele lines, we observe no signal or very weak signal for GST. This meaning that what we see in the RNA-Seq and the 3' Seq is actually real. Furthermore, we characterize, in order to identify if the SNP is actually the causative SNP, as Nacho showed, we looked at we did a luciferase reporter assay, and we clone it, the enhancer in front that regulates the expression of the luciferase assay. And you can see that the major allele is higher than the minor allele, or the promoter region of the GST is from the 2 different genotype is different and in the same direction as detected by the Tag-Seq. In order to prove that this is the causal, we did the exact same experiment that Nacho explained before. So we substituted the causative SNP from the minor to the major and vice versa. And what we notice is that this single nucleotide variation is in linkage disequilibrium with the structural variation that in this line is a little bit shifted. And that's why we then call as major structure, like major causes, because there are slightly shifted, this variation has slightly shifted, so it's not common to all lines. But what we see in with this experiment is like the SNP that is in linkage with the structural variation is actually the causative SNP, because when we substitute the major allele into the minor background, what we see is that we don't have expression at all, while we have a small expression in the major, in the vice versa experiment. And now what we notice, okay. So what we did is, furthermore, in order to observe where these EQTLs are located, we observed that these EQTLs are located mostly at the 5' end of the gene, like where promoter elements or proximal elements, regulatory elements of the transcription are located. And what we see, and actually this is what Dermot did, another PhD in his lab, what Dermot did is to calculate the enrichment in different regulatory elements defined by DNase, SA, DNA-Seq, ChIP-Seq, or simply a transcription factor binding site that are bound in ChIP-Seq. And what we see is that there is an enrichment for both tissue-specific or stage-specific and common EQTLs. Okay. And here is just an example of what we see, and this is a case for the gene that is CG10306, where there is a single nucleotide polymorphism that affects transcription factor binding site. That is a transcription factor binding site for sloppy pair. And we see that sloppy pair, ChIP-Seq show occupancy here of the transcription factor exactly in this region. And as you can see on the change in expression plot, on this box plot, you see that there are significant differences. There is an increase in the minor alleles in comparison to the major allele. And this is in concordance with the role of sloppy pair that is a repressor. So in order to see if this was actually the causative SNP or the causative element, what we did is again luciferase assay in Drosophila S2 cells. And fortunately, the S2 cells do not express sloppy pair, so we had to co-transfect it together with the control and enhancer vectors. And what we see is when we have absence of sloppy pair, there are no differences in the expression of the three alleles that we have, major, minor, and the mutation of the major SNP into the minor SNP, so that we have the minor allele into the major background. But when we add sloppy pair, we see a strong repression almost to the we have a strong repression of the major allele, and we observe a significant derepression of the minor allele. But what is interesting is that when we mutate the causative SNP, so when we mutate this T in the major allele into the C of the minor allele, and only this mutation, we observe a stronger repression of the expression of the luciferase assay. So we start thinking that there was some kind of interaction or allelic interaction between SNPs within the regulatory elements. And for this reason, what we did is to test many other elements, and here you can see in this plot, and we observed that there is definitely a allelic interaction within the regulatory elements that reduce the effect of, or that buffer the effect of the SNP or the single nucleotide polymorphism or structural variation on the phenotypic level. So, and this is one thing that we didn't expect. So just to now that we show this, what we wanted to do is to take advantage of the technique and look at those EVTs that are affecting specifically the 3' polyadenylation or the 3' ends of the transcripts. And we call these EQTLs 3I QTLs. We identify a bunch of them that are common and stage specific in a bunch of transcription, in a bunch of factors as well that are important for the lethality and the viability of the organism. But since we have a single nucleotide resolution of the transcription end site, what we wanted to do is to look at the motifs that are around this transcription end site. And not surprising, what we detect are a motif for the polyadenylation, the classical PAS motif, AATAA, but we call also motifs that are upstream and downstream canonical motif, in particular these two. We call also motifs that were previously identified in another study, but surprisingly, since we sequence 80 lines, you can say that we have a huge depth in sequencing of the 3', and we call also some other motifs, some new motifs that were not described before, with some positional enrichment. Or location, sorry, position like with a precise location in comparison to the TES, the transcription end site. So what we did afterwards, so we call these 3 EQTLs, that are those EQTLs that affect the 3' polyadenylation end. And what we saw is like these 3 EQTLs are specifically enriched in motifs that are characteristic of the polyadenylation site, but not only de novo and or old already known, but also some other elements that are involved in the 3' stabilization and regularization of stabilization of the 3' end or degradation, or inducing degradation of the transcript, binding the 3' end of the transcript itself. And this is the case of ELAV, but what is surprising is this TRA2. TRA2 is a gene that is involved in splicing, but what we see is actually something interesting. So we observed that there is a clear, like, specific effect site that correlates with a specific phenotype. In particular, if the SNP creates or break the TRA2 motif at the 3' end, we observe an increase or a decrease of the usage of that specific alternative polyadenylation sites. And this is not observed when the TRA motif is in splicing site. This is actually very correlated with the PAS motif, canonical PAS motif, where we observe as well differences in usage of the polyadenylation, depending if the PAS motif is created or broken. Now, and let me show you an example that we validated. So as usual, you see the single nucleotide polymorphism that is identified, that is highly significant in this locus for C1. That is the specific gene. You see the coverage from the major allele and the minor allele, and as well the mean in the row number normalized reads of the RNAs of the 3' Tag-Seq in this locus. And what you can see is that there is this particular polyadenylation site that is used by the minor allele only and not by the major allele. This is because there is a SNP that creates an AATAA site that is a canonical PAS polyadenylation site recognized by the polyadenylation factors. And when we test this in luciferase assay, so we clone this 3' end or 3' UTR at the end of the CDS of the luciferase, we observe that there is, depending on if we have a major or a minor allele, we have an increase or decrease of the stability of the RNA in luciferase SA. So this meaning that what we are calling is a causative SNP. And why I'm saying that this is a causative SNP, because in the region that we clone at the 3' end of the luciferase SA, this was the only SNP that was present. Now, so another thing that we did, and thanks to the 3' Tag-Seq that allow us to do it, was to look for EQTLs that change the length of the 3' UTR. Because you can see, like in this case, you have an extension of the 3' end when you create these polydengylation sites. So we started to do this genome-wide, and we identify a significant, like around 300 of these 3' UTR QTLs. And there are UTLs that increase the length of, there are UTR QTLs that increase the length of the 3' UTR, and there are UTR QTLs instead that decrease the length of the 3' UTR. And you can see this in this plot, where in purple you see the extension, the lengthening, or the shortening in green of the 3' UTR. And here, like in the next slide, you will see two examples of this, where you can see like the HF as shown here, that as a shortening of the 3' UTR going in this direction, as you can see here, where you have another polydengylation site in here that is not present in the minor allele. This is because the polydengylation signal, the canonical AATAA, is broken by a SNP. And we test this as previously described in luciferase SA, this time changing the major allele into the minor allele, the major SNP into the minor SNP, and what we see is that there is a decrease in the activity in the stability or in the production of the luciferase in vitro in S2 cells. While in case of the YL1, we have an extension of the 3' UTR, because in this region we have, specific mutation of the polydengylation site that is destroyed. So in this case, the polymerase continues to transcribe until it finds another downstream, a more distal polydengylation site. And so in conclusion, we can say that at the end, what we describe is that there are QTLs that affect a large number of essential developmental genes. In particular, we observe that 17% of the genes that are expressed in embryonic development are affected by the development by QTLs. We have QTLs that are affecting both transcriptional and the post-transcriptional process, those QTLs that are located in regulatory elements, and those QTLs that are located at the 3' end of the transcript. And we show also that there are some allelic interaction amongst variants in developmental enhancer that partially buffer each other effects. And with this, I would like to acknowledge Eileen in